Chapter 12, Section 4, Mutations. Now and then, cells make mistakes in copying their own DNA, inserting an incorrect base or even skipping a base as the new strand is put together. These mistakes are called mutations, from the Latin word meaning to change. Mutations are changes in the genetic material. Kinds of mutations. Like the mistakes that people make in their daily lives, mutations come in many shapes and sizes. Mutations that produce changes in a single gene are known as gene mutations. Those that produce changes in the whole chromosome are known as chromosomal mutations. Gene mutations. Gene mutations involving changes in one or a few nucleotides are known as point mutations, because they occur at a single point in the DNA sequence. Point mutations include substitutions, in which one base is changed to another, as well as insertions and deletions, in which a base is inserted or removed from the DNA sequence. Substitutions usually affect no more than a single amino acid. The effects of insertions or deletions can be much more dramatic. Remember that the genetic code is read in 3 base codons. If a nucleotide is added or deleted, the bases are still read in groups of 3, but now those groupings are shifted for every codon that follows, as shown in Figure 12-20. Changes like these are called frame shift mutations because they shift the reading frame of the genetic message. By shifting the reading frame shift mutations may change every amino acid that follows the point of the mutation. Frame shift mutations can alter a protein so much that it is unable to perform its normal job or function. Figure 12-20. Gene mutations result from changes in a single gene. These diagrams illustrate the different types of mutations or changes in DNA. They also show how the mutations affect the amino acid sequence of the proteins for which they code. In a substitution, on the left, one base replaces another. In an insertion, center of diagram, an extra base is inserted into a base sequence. The loss of a single letter in a sentence below models the effect of the deletion of one base in a DNA sequence. Chromosomal mutations. Chromosomal mutations involve changes in the number or structure of chromosomes. Such mutations may change the location of genes on chromosomes and may even change the number of copies of some genes. Figure 12-21 shows 4 types of chromosomal mutations: deletions, duplications, inversions, and translocations. Deletions involve the loss of all or part of a chromosome, while duplications produce extra copies of parts of a chromosome. Inversions reverse the direction of parts of chromosomes, and translocations occur when part of one chromosome breaks off and attaches to another. Significance of mutations. Many, if not most, mutations are neutral, meaning that they have little or no effect on the expression of genes or the function of the proteins for which they code. Mutations that cause dramatic changes in protein structure or gene activity are often harmful, producing defective proteins that disrupt normal biological activities. However, mutations are also the source of genetic variability in a species. Some of these variations may be highly beneficial. Mutations are the causes of many genetic disorders, including sickle cell disease and cystic fibrosis, both discussed in Chapter 14. Harmful mutations are also associated with many types of cancers. In contrast, beneficial mutations may produce proteins with a new or altered activity that can be useful in changing environments. One such mutation produces resistance to HIV, the virus that causes AIDS. Plants and animal breeders often take advantage of such beneficial mutations. For example, when a complete set of chromosomes fails to separate during meiosis, the gametes that result may produce triploid or tetraploid organisms. The condition in which an organism has extra sets of chromosomes is called polyploidy. Polyploid plants are often larger and stronger than diploid plants. Important crop plants have been produced in this way, including bananas and many citrus fruits.